All right, let's get into it. Today, we're putting on our detective hats and diving into the world of genetics. We're going to find out how scientists can pinpoint one single, totally invisible genetic clue that's buried in a sea of millions of others. This is a technique that's absolutely vital for everything, from spotting diseases early to making sure the food we eat is safe.
So just imagine this for a second. You've got a biological sample, right? It's basically a chaotic soup of genetic stuff. And your mission, should you choose to accept it, is to find the one tiny microscopic signature of a specific virus. Seriously, how on earth do you find that single genetic needle in a haystack the size of a mountain?
Well, the answer, and really the hero of our story today, is a brilliant technique called qPCR. It's the amazing tool that lets scientists not only find that needle, but to actually count how many of them there are.
To really get why this is such a game changer, we first have to appreciate the problem. Whether a doctor is diagnosing a disease, a researcher is studying cancer, or a lab is testing food, they all need a way to detect incredibly tiny amounts of specific DNA or RNA.
So how does qPCR actually pull this off? Honestly, the best way to think about it is like a super-powered DNA photocopier, but with this really cool twist. It doesn't just make billions of copies of the gene you're looking for, it makes every single copy glow, and it counts them in real time as they pop up.
Let's just break down that name for a second. qPCR stands for Quantitative Polymerase Chain Reaction. The polymerase chain reaction, or PCR part, that's the photocopier. It's the engine that makes billions of copies of a specific bit of DNA. And the quantitative part, that's where the magic really happens. We add fluorescence, basically a glowing signal, to measure the amount of DNA as it's being copied. So it's just copying plus glowing, that's the whole idea.
Okay, so how do we actually make the DNA glow? Well, there are two main ways to do it. The first one uses a dye called SYBR green. You can think of this like a big floodlight. It's simple, it's cheap, but it lights up any double-stranded DNA it bumps into. The second way, though, is much more like a laser pointer. It uses a custom-designed probe that will only bind to and light up the exact gene you're hunting for, which makes it way more precise. So, we've got potentially billions of glowing copies of our DNA. But how do we turn all of that light into data that actually means something? How is that signal even read by the machine?
The answer comes down to the single most important number in any qPCR experiment, the cycle threshold, or CT value. See, the machine copies the DNA in cycles, 1, 2, 4, 8 copies, and so on. The CT value is simply the number of the cycle when the glow finally gets bright enough to cross a detection line. And here's the key thing to get. What matters isn't how bright the light gets at the end, but how early it shows up.
So for instance, a low CT value, let's say 18, means the glow was detected really early on. And that tells you that your original sample must have been jam-packed with the target DNA you were looking for. It just didn't take very many copying cycles to get a really strong signal.
Now compare that to a high CT value, something like 36. If you see that, it means the glow appeared much, much later in the process. And that tells you there was only a tiny amount of your target DNA to start with. So the big takeaway here is that CT values work backwards. A low number means a high amount of your target. Low CT, lots of stuff. High CT, not so much stuff. It's really that simple.
Okay, so we have a CT value. We have a good idea of how much signal there is. But this brings up the next big question for any good detective. Is this signal even real? How do we know for sure that we copied the right gene and not just some random genetic junk?
This is where another really clever feature comes into play, the MELT curve analysis. It's basically a quality control check at the very end of the experiment. The machine slowly heats up all the DNA it just made, and because every unique piece of DNA has its own specific melting temperature, the machine can tell if you made just one pure product or a messy mix of different things. It's pretty much the machine's built-in lie detector.
And this is what you want to see. One clean, sharp peak. A single peak like this is your confirmation, your proof that the reaction worked perfectly and that your DNA photocopier only copied the one gene you're interested in. This is the green light for your data.
But then you might see something like this. Seeing multiple peaks is a huge red flag. It means your photocopier kind of went rogue and started copying other stuff you didn't want, like little bits of genetic junk we call primer dimers.
If you see a messy curve like this, your CT value is basically useless, because that glow is coming from more than just your target.
So let's say you've got a great CT value, and you've confirmed your signal is real with a perfect MEL curve. Now you can finally get to the ultimate goal for so many scientists, comparing different samples to figure out what our genes are actually doing.
Now, the method for this sounds really complicated, but the logic behind it is actually pretty straightforward. First, you need a reliable reference point. In genetics, we use what's called a housekeeping gene. It's a stable gene that's pretty much always on and doesn't change much. That's our baseline. We measure our target gene, then we measure the housekeeping gene, and we do this for both our experimental sample and a normal control sample. By comparing all those numbers, we can calculate the final result, which is called the fold change.
And that fold change, that's the answer you've been working for. It tells you exactly how much a gene's activity has changed in your sample. For example, you can see if a certain gene in a cancer cell is working 10 times harder than in a healthy cell. We call that upregulation. Or maybe it's been almost completely turned off. That's downregulation. This is how we start to uncover the secrets of how diseases work at a genetic level.
And look, this isn't just some abstract technique that happens in a lab. This is a tool that has completely changed how we see the biological world around us. It's incredibly fast, it's super sensitive, and it is unbelievably powerful.
The applications are just everywhere. It's the technology behind the rapid detection of viruses like the flu or SARS-CoV-2. It's absolutely essential in cancer research for figuring out which genes are making tumors grow. It's used all the time in medical diagnostics, in making sure our food is safe to eat, and so much more. This is science that truly impacts all of our lives.
So, where does that leave us? Well, from finding that one viral gene in a million to decoding the complex dance of our own genes, qPCR has given us this incredible window into our biology. And what's so exciting is that the technology is getting better all the time. So you have to wonder, as it gets even faster, even more precise, what incredible secrets are our genes going to reveal to us next?